Hello guys, welcome to my channel. So as you know, we are studying about the protein function, and basically we studied about the hemoglobin, that how it carries and transport the oxygen. So we are just going through that as well and continuing regarding the hemoglobin that binds the oxygen. So the hemoglobin must bind the oxygen efficiently in the lungs, where the partial pressure of the oxygen should be about 13.3, right? So that's the kilopascal unit, right? And so the in lung, when the hemoglobin bind the oxygen efficiently, the partial pressure of oxygen should be about 13.3 kilopascal per unit, and releases oxygen in the tissue. And the tissue, so this is for lungs, and the partial pressure for the tissue is 4 kilopascal, right? So that's the partial pressure of oxygen in the lungs, and that's the partial pressure of oxygen in the tissues, right? Right. So the protein that is binding to the oxygen, that is bound to the oxygen. So like hemoglobin is also binding to the oxygen, myoglobin is also binding to the oxygen, and there are some other proteins as well. But the protein that bound to the oxygen with the high affinity would bind it efficiently in the lungs as well, right? But would not release much of it in the tissues. So there is a difference. So if the protein bound oxygen with a sufficiently low affinity to release it in the tissues, it would not pick up much oxygen in the lungs, right? So the whichever protein that is bound to the oxygen, if the affinity is low to release it in the tissue, then for the pickup as well, right? So the it would not pick up the oxygen in the lungs too. So this problem is solved by the hemoglobin protein. So hemoglobin solves this problem. How it solves this problem? Right, so hemoglobin solves this problem, and how it solves this problem? By undergoing a transition from a low affinity state, right? So the last time we studied about this. So low affinity state, that is the T state, to the high affinity state, there is R, or I will write it down here, R state. All right. Right. So hemoglobin, how hemoglobin solves this problem? So as we know, hemoglobin is a multi-subunit protein, right? But like for example, if there is like just a single subunit protein, so it cannot provide that high affinity or the low affinity, because when the ligand binding site of the single subunit, when it binds, so each molecules of the ligand binds independently and cannot affect the ligand binding site on another molecule. But in contrast, for the hemoglobin, when the oxygen binding to individual subunits of hemoglobin can alter the affinity of for oxygen in the adjacent subunits. So how is it? When the first molecule of oxygen that interacts with the deoxyhemoglobin binds weakly. So for example, let's see. So like one, two, for example, this is like, I'm just giving the example. Like this is different subunits for hemoglobin. So now it does not have any oxygen right now. So that's why it is deoxyhemoglobin. It is deoxyhemoglobin. So the first molecule of oxygen, so that will bind, for example, here, O2, right? That will bind weakly because this will bind to a subunit that is in the T state. And T state has low affinity state for the oxygen. So when it is deoxyhemoglobin, when the hemoglobin does not have any of the oxygen, so when the first oxygen that binds with the first subunit, that will be in the T state, so it binds weakly. And it's this binding, however, leads to conformational changes that are communicated to the adjacent subunits, making it easier for additional molecule of oxygen to bind. So when this binds to this, then it will make a conformational changes to this, for example, and this will like whatever, just for an example, it will change, it will have conformational changes to this different subunits, and then this will bind perfectly as it will change to the high affinity from the T state to the T state to the R state, right? Right, so the last molecule, that is that oxygen molecule just binds to the heme in a subunit, right? This oxygen are binding to the heme in the subunit of the hemoglobin. So that is, and the last molecule, for example, this is the last molecule, and that is binding to the heme subunit. So this will be already in the R state, and hence it binds with much higher affinity than the first molecule. So this type of binding, that is represented, and that is known as allosteric protein. What is known as allosteric protein? So this means allosteric proteins, that means in which the binding of a ligand to one side, that will affect the binding properties of another side on the same protein, right? And the term this allosteric is derived from the Greek. So allos means, so this is term is derived from the Greek. Allos means other, and stereos means shape, that is other shape, right? Or it's also solid or shape, right? So these allosteric proteins are those having other shapes or conformation induced by binding of the ligands, and that is referred to as a modulators. So these ligands are referred to as modulators. All right, so this hemoglobin is allosteric protein. Why it's allosteric protein? Because the ligand that is binding to one side, that affects the binding properties of the another side on the same protein, right? And the conformational changes induced by the modulator that interconvert more active and less active forms of the protein. And the modulators of this, for this allosteric protein, may be either an inhibitors or activators, right? Right, and when the normal ligand and modulators are identical, the interaction is termed as homotropic. And when the modulator is a molecule other than the normal ligand, the interaction is heterotropic. So some proteins have two or more modulators and therefore can be both homotropic and heterotropic interactions. All right, so hemoglobin transports oxygen, but it does not only transport oxygen. Hemoglobin hemoglobin also transports, what it transport? H plus, that is proton, and CO2, that is carbon dioxide, right? So in addition to carrying nearly all the oxygen required by the cells from the lungs to the tissues, hemoglobin carries these two end products of the cellular respiration. So these end products are of the cellular respiration, that is the proton and the carbon dioxide, from the tissues to the lungs and the kidneys, where these are excreted, right? So the carbon dioxide is produced by the oxidation of organic fuels in mitochondria, is hydrated to form bicarbonate. So this, what happens to this? It's CO2 oxidation and reverse reaction, right? This will form the bicarbonate product. Right, so this reaction is catalyzed by carbonic anhydrase. So that's an enzyme. Oops. Right. Anhydrase, right? So this is an enzyme particularly abundant in erythrocytes, right? So the carbon dioxide is not very soluble in the aqueous solution. CO2 is not very soluble in water, and the bubbles of this carbon dioxide would form in the tissues and blood if it were not converted to bicarbonate, right? As you can see, the carbon dioxide when you drink the sodas, right? Sparkling waters. So the carbon dioxide is not very soluble in this aqueous solution, and so if this carbon dioxide is not converted to this bicarbonate, it might form bubbles in your tissues and in the blood. So that's why it's converted into this bicarbonate. So you can see from this reaction that this reaction is catalyzed by this enzyme carbonic anhydrase, right? And you can see the hydration of this carbon dioxide that will result in also in H plus, that is protons concentration. So that means it will decrease the pH in the tissues. So the binding of oxygen by hemoglobin is profoundly influenced by the pH and the CO2 concentration. So the interconversion of CO2 to bicarbonate and bicarbonate to CO2 is of great importance to the regulation of oxygen binding and release in the blood. So hemoglobin transports about 40% of, so it transports about 40% of protons and 20% of, and 15 to 20% of, 15 to 20% of carbon dioxide, right? That is formed in the tissues. So the remainder of the H plus, this H plus is actually absorbed by the plasma bicarbonate buffer, and the remainder of the carbon dioxide that is transported as dissolved as the bicarbonate. So the binding of this H plus, that's proton, and the carbon dioxide is inversely related to the binding of oxygen, right? Right. So that is important. So the binding of H plus and the CO2 is inversely related to the binding of oxygen. So that means at the relatively low pH and high concentration of CO2 in the peripheral tissues, the affinity of hemoglobin for oxygen decreases as the H plus and the CO2 are bound and the O2 is released into the tissues. Conversely, in the capillaries of the lungs, as the carbon dioxide concentration is, or the CO2 is excreted and the blood pH consequently rises, the affinity of hemoglobin for oxygen increases and protein binds more oxygen for transport to the peripheral tissues. Right, so we'll explain again just with the simple diagram. All right, for example, let's see, this is hemoglobin that has oxygen attached, and this is going into the tissues, right? So we'll do traveling into the tissues, and for example, this is this tissues. So there is, so this tissues has low pH and high CO2, high carbon dioxide concentration. So the affinity of hemoglobin for oxygen will decrease. So when this hemoglobin comes here, the affinity of hemoglobin will the affinity of hemoglobin for the oxygen will decrease. We'll write it down here. Affinity will decrease for oxygen. So how it will decrease? As this H plus, this proton, and so at this stage, for example, when it here comes here, so it will release oxygen, and this is hemoglobin, and attaches plus and CO2. So the affinity decreases for oxygen, so it will release oxygen in the tissues, and the H plus and the CO2 will bind, right? So in the same way, when this, so now hemoglobin has this, so hemoglobin is traveling through lungs, right? So in the capillaries of lungs, right? So lungs, capillaries. So this is capillaries. I will write it down here. Lung capillaries of lung. This carbon dioxide is excreted in here. So when this hemoglobin comes here, CO2 is excreted, right? And the blood pH will consequently rise, right? And the blood pH will increase, consequently rise. So that will have the affinity of hemoglobin for oxygen increases, and the protein binds more oxygen for transport to the peripheral tissues. So that's just a simple. So this effect of this pH and the carbon dioxide concentration on binding and release of oxygen by hemoglobin is known as Bohr effect. All right. So right. So this effect, the effect that we see, this effect of pH and CO2 concentration on the binding and release of oxygen, that is by hemoglobin, is called as the Bohr effect. Right, so this is the Bohr effect, and this is named after the Christian Bohr, the Danish. This is the Danish physiologist, right, who discovered this in the 1904. Right, so both the oxygen and the proton H plus are bound by the hemoglobin, but with inverse affinity. So when the oxygen concentration is high, as in the lungs, hemoglobin binds oxygen and releases protons. But when the oxygen concentration is low, as in the peripheral tissues, proton is bound and oxygen is released. So that means, so the how the reaction we can write it down like this, plus oxygen. This is reversible reaction, plus H plus. All right. So this is because of the proton. All right, and the oxygen and the proton are not bound at the same site in the hemoglobin. So we'll just go through a bit of this oxygen binding on a molecular level or biochemistry level that we are studying. So oxygen and the protons are not bound at the same sites in the hemoglobin. Oxygen binds to the iron atoms, right, of the heme, whereas the proton binds to any of the several amino acid residues in the protein. But this major contribution to the Bohr effect is made by the histidine. It's made by this histidine 146, or it's also known as histidine HC3. Right, so the oxygen and the hydrogen H plus are not bound at the same sites in the hemoglobin. So the oxygen binds to the iron atoms of the heme, whereas the proton binds to any of the several amino acid residues in the protein. So the major concentration of this Bohr effect is made by this histidine 146, that's also known as histidine HC3 of the beta subunit, And when the hemoglobin is protonated, this residue forms one of the ion pair to ASP94, that is aspartic acid. So this ASP is the aspartic acid amino acid, and that is, we can write that down as ASP FGI. Alright, so this will help stabilizes the deoxyhemoglobin in the T state. So this ion pair will stabilizes the deoxyhemoglobin in the T state. Right, so this ion pair stabilizes the protonated form of histidine HC3. This will giving this residue an abnormally high pKa. So that's the ionization constant, pKa. So this is the ionization constant in this is in the T state, and when this ionization constant falls to its normal value of 6 in the R state, because the ion pair cannot form and this residue is largely unprotonated in the oxyhemoglobin at pH level 7.6. So the blood pH in the lungs, right? So as the concentration of proton rises, protonation of histidine HC3 promotes release of oxygen by favoring a transition to the T state, and the protonation of amino terminal residues of the alpha subunit, certain other histidine residues and perhaps other groups has a similar effect. Thus we see that the four polypeptide chains of the hemoglobin communicate with each other, not only about the oxygen binding to their heme group, but also about the H plus that should binding to the specific amino acid residue. And hemoglobin also bind carbon dioxide again in a manner inversely related to the binding of oxygen. So that is the carbon dioxide bind as a carbamide group, right? CO2 bind as a carbamide group. So bind as a carbamide group, right? This will carbamide group to the alpha amino group at the amino terminal end of each globin chain, forming carboamino hemoglobin. All right, and there is one more thing. So the oxygen binding to hemoglobin is also regulated by 2,3 biphosphoglycerate. All right, so let's remove this. So the oxygen binding to the hemoglobin is also regulated by 2,3 biphosphoglycerate. So this interaction of 2,3 biphosphoglycerate, so that's in short also known as BPG, with hemoglobin molecules further refines the function of hemoglobins and also provide that it is an heterotropic allosteric modulation. So on your screen you can see the picture of this 2,3 biphosphoglycerate. So this BPG is present in relatively high concentration in erythrocytes. So this is present in high concentration in erythrocytes, and when hemoglobin is isolated, it contains substantial amount of bound BPG, which can be difficult to remove completely. So this 2,3 biphosphoglycerate is known to greatly reduce the affinity of hemoglobin for oxygen. So there is an inverse relation between the binding of oxygen and the binding of BPG. So we can say that like this, the reaction, this is hemoglobin and BPG together and the oxygen binding, and this is reversible, right? Right, so this is, okay, wait. This is like this plus the BPG. Right, so this 2,3 biphosphoglycerate, what it reduces the affinity of hemoglobin for oxygen. So that why the reaction is its inverse relationship between the oxygen and the binding of BPG. So this BPG bind at the site distance from the oxygen binding and regulates the oxygen binding affinity of hemoglobin in relation to the partial pressure in the lungs. So this binds to a site distance from the oxygen binding site and it regulates the oxygen binding affinity. What it do? It regulates O2 binding, regulates the O2 binding affinity of hemoglobin in relation to the partial pressure of oxygen in the lungs. So that's why this BPG is important in the physiological adaptation to the lower partial pressure of oxygen at high altitudes. Why? So like for a healthy human at a sea level, the binding of oxygen to hemoglobin is regulated such that the amount of oxygen that is delivered to the tissue is nearly 40% of the maximum that could be carried by the blood, right? So imagine that this person is suddenly transported from the sea level to an altitude of like 4500 meters where the partial pressure of oxygen is considerably lower. So at that time the delivery of oxygen to the tissue is now reduced. So what happened after a few hours at a high altitude, this BPG concentration in the blood has begun to rise, leading to the decrease in the affinity of hemoglobin for oxygen. So this adjustment in the BPG level has only a small effect on the binding of oxygen in the lungs, but a considerable effect on the release of oxygen in the tissues. So as a result, because of this, the delivery of oxygen to the tissues is restored to nearly 40% of the oxygen that can be transported by the blood. Right? So I hope you got the point. So when this BPG level rises in the blood, so this BPG level has the affinity, it decreases the affinity of hemoglobin for the oxygen. This BPG level has only a small effect on the binding of oxygen in the lungs, but it has a considerable effect on the release of oxygen in the tissues. So as a result, so when this hemoglobin reaches to the tissues, so there is the high BPG concentration. So the more BPG will get attached to the hemoglobin and the oxygen will be released. So that is how it is the 40% for the tissue is restored like this. You got it? So the situation is reversed when the person returned to the sea level, and this BPG concentration in erythrocytes also increases in people suffering from hypoxia, leading to hypoxia, that is lowered oxygenation of peripheral tissues due to inadequate functioning of lungs or circulatory system, right? Right, and this regulation of oxygen binding to hemoglobin by BPG has an important role in the fetal development because a fetus must extract oxygen, right, from its mother's blood. So the fetal hemoglobin must have a greater affinity than the maternal hemoglobin for the oxygen. So the fetus synthesis gamma subunit rather than beta subunits, forming like the fetal has alpha 2 and gamma 2 hemoglobin instead of alpha 2 beta 2. So this tetramer has much lower affinity for BPG than the normal adult hemoglobin and correspondingly higher affinity for the oxygen. Alright guys, so that was the primary step for the protein function, that is just for the all the globins that transports the oxygen or to storage the oxygen, right? So this is the first level and we'll see you. Alright guys, so we'll see you in the next video. Alright, thank you guys for watching and don't forget to subscribe the channel.